This is a recording for Biology 1406. The subject is the chemistry of carbon, the last of the study material for Unit 1, exam number 1. So the chemistry of carbon is important because after water, you are primarily composed of organic molecules based on carbon. So this is the chemistry of carbon. Molecules with 2 or more carbons in them traditionally are referred to as organic molecules. And so when we say organic molecules, we're inevitably referring to something whose core is made out of carbon. And we want to identify why carbon occupies that role, how it does that, and practically, importantly throughout the semester, the chemical modifications to carbon chemistry that are caused by functional groups attached to those carbon molecules. So you will recall that your molecular composition is primarily carbon, hydrogen, oxygen, and nitrogen. And the carbon molecules in you are primarily composed of carbon and hydrogen, or carbon, oxygen, and hydrogen, and then less frequently carbon, hydrogen, nitrogen, sulfur, and other things like that attached to them. And that's largely a role of functional groups. So let's go ahead and talk about carbon a little bit. And I'm using the PowerPoint for the last portion of Chapter 2 in your OpenStax textbook. You can use any biology reference you want. The Campbell book certainly covers this material at least as well as your OpenStax book. All right. So we're doing this because in the next chapter, on the first chapter of Unit 2, we'll take a look at the macromolecules that are formed by carbon, and those of interest in living things. All living things are proteins, carbohydrates, lipids, and nucleic acids. 4 major classes of organic macromolecules. And macro because these then have some other chemical features in them which allow them to form truly, truly huge molecules from a chemical standpoint. Now, one of the reasons carbon is so good at doing this and ends up being the core of organic chemistry is because not many atoms can do this, which is to form up to 4 covalent bonds with other atoms. So carbon allows you to build skeletons, shapes that are really, really quite diverse. And that's what we mean by the carbon backbone of an organic molecule or of a macromolecule. All right. And that is because the 4 bonds is due to the fact that each of them has 4 electrons in their outer or valence shell, and that's shell number 2 for carbon atoms. And there are 4 electrons in that outer shell, and that means there are 4 possible sources of electrons to finish filling that shell, and that can be up to 4 other covalent bonds. All right, satisfying that octet rule of having 8 electrons in the outer shell after the very first shell, which only allows 2. All right. The simplest organic molecule is what we refer to as a hydrocarbon, and the name says it all: hydrogen and carbon, hydrocarbon. This is also in the world of, chemical engineers and stuff like that, the name for almost all of our fuels. The bulk of the things that we burn, combust for fuel, are combinations of carbon and hydrogen. So methane, ethane, butane, gasoline, diesel, propane, all of those liquid fuels, almost all of them that I can think of, are hydrocarbons. So they're some form of a carbon and hydrogen skeleton. And they store a huge amount of energy because the bond between a carbon and a carbon, and more practically a carbon and a hydrogen, is referred to as a high energy bond. It is a nonpolar bond. Both of these atoms are equally electronegative, so electrons that are present in a covalent bond between them are shared equally between the 2 atoms, and they're equidistant from both nuclei, if you will. So they're not close to a nucleus, they're kind of far away from both nuclei. So if you can yank those electrons off and give them to something that will pull them closer, like oxygen will, you can get those electrons to release a huge amount of energy, and that is exactly what we do when we burn a fuel. Those high energy electrons with the hydrogens attached to them are yanked off of the carbon molecule and are coupled with oxygen to make water, which has a much lower energy level. It releases all that energy as heat and light, which we use. Chemically, you and every other living organism on Earth uses the same sort of idea to power itself. We don't release flames and lots of heat inside our cells. We capture the energy chemically, but we break down things like hydrocarbons, including hydrocarbons. We personally can't use hydrocarbons, but lots of living things can. And extract energy from them and produce water as a byproduct and carbon dioxide as a byproduct, just like a flame. So that's one of the reasons, organic molecules and carbon chemistry are so important to us, because of this ability to store energy. All right, so let's take a look at the simplest of carbon molecules, and that's methane, which has a central carbon. And remember, carbon can bind to up to 4 different things. So in this particular case, it's bound to 4 electrons right here. For the record, if you're not familiar with chemistry notation, the straight line represents a covalent bond. And in this sort of drawing format, the wedge-shaped line means this is coming out of the sheet of paper towards you. And the dotted line right here means the hydrogen would be behind the sheet of paper going back. And it's an attempt to recreate the 3-dimensional structure of this thing on a 2-dimensional sheet of paper. So anyway, that shape is called a tetrahedron. So we can bind one carbon to another. All right. So here are some hydrocarbons. All right. So here's that simple one. It's a tetrahedron with just a single carbon at the center right here. The molecule is methane, CH4. You should get to know that one. Another one that we use, to as a fuel source is ethane right here, and as a solvent. And it has 2 carbons right here. And remember, each carbon is going to make 4 bonds. So it's bound to 1, 2, 3, 4 other things. Just the 4th thing now is another carbon. So a single covalent bond between these carbons, and then each of them, to finish its octet rule, has got 3 hydrogens attached to it. There's your basic rule. If I wanted to do 3 carbons in a row, I would make 3 carbons in a row, add 3 hydrogens to 1 carbon. The middle carbon, which would be in here in this picture, would have a hydrogen on either side of it. And the last carbon at the other end would have 3 hydrogens, hanging off of it. So that hydrocarbon with 3 carbons in it, again, each carbon would be bound to 4 other things, and that's the important idea here. Now, you can also make double bonds or rarely even triple bonds. So this is a double bond between 2 carbon molecules, and the significance of this is now the carbons are only going to bind to 2 other hydrogens right here, because 1, 2, 3, 4 bonds, that's all they can make. And also, importantly, this carbon can't rotate relative to this one, because they're fixed by these 2 bonds right here. Single bonds can spin and rotate. All of these bonds, you can take the atom at the end and rotate it, if you will, on that single covalent bond, and nothing cares. So here's some examples of some other structures that can be made with carbon. These are called aromatic rings. And there's cyclopentane right there. Here's cyclohexane, hex for 6. Penta, like the pentagon for 5. And notice again, all the hydrogens attached right here to satisfy their valence chemistry. Every one of these has got 4 bonds. Same thing right here. Here's an interesting one where we've got these 3, and this is the aromatic ring properly right here, because it vaporizes into a gas quite easily. Anyway, this has 3 triple bonds in it right here, and notice, therefore, we've only got single hydrogens to make the 1, 2, 3, 4th bond, for each carbon right here. This is benzene, which is quite a toxic component, traditionally of diesel fuel. We try our darndest to keep that out of those sort of fuel supplies these days, because it's a very toxic compound. And here's an example of that molecule modified by putting a nitrogen into this spot right here. And that's kind of the intro to the whole idea of organic molecules and life, is that we can take these basic carbon structures, and then modify them by replacing carbons and replacing hydrogens with other things. All right. We need to get this idea of what's called isomers, which just means same formula in this particular case. So there are molecules that have the same formula but different arrangement. All right. Some molecules are so simple there's only one arrangement, for the atoms that make it up. But once you get a couple of carbons in there, there's lots of different ways you can organize them. So for instance, right here we've got a 4-carbon molecule. And here we have a 4-carbon molecule. This one's branched. It has a line going this way and then a branch with the carbon going off of here. And I could replace this hydrogen with another carbon and do that endlessly to make however big molecule I wanted. The important idea here is that both of these have the same sort of, they have the same exact chemical formula, but very, very different shapes and theoretically different functions in living organisms if you were to use them. All right, because of that branch versus a straight chain here. And if you need proof, count up your hydrogens that they're the same formula. 8, 9, 10, 3, 6, 7, 8, 9, 10. All right. Then we have these things called, these are structural isomers right here. They just have a different shape. Okay, they have a different shape, but same formula. These are structural isomers. They still have the same formula, but they have different shapes because of the presence of a double bond here. Remember, the double bond can't rotate. So here we've got the what are called methyl groups on the same side of the double bond. That's what we refer to as a cis bond, a cis double carbon bond. You'll see this term more. And here's a trans double bond, all right, where the double bond, the methyl groups are on opposite sides of the double bond. And this can't be switched to this without breaking a bond. We have to break bonds to rebuild this in this form and vice versa. So that's what we mean by a geometric isomer. And then finally, the most unusual version of this, enantiomers, which are left and right-handed molecules. So they are mirror images of each other, and one can't be converted into the other. So if you play around with this idea enough, try and visualize it, you'll see that this and this are mirror images of each other, but therefore not identical. They are left and right versions. And we use that term, left, L for linkstrous, the Latin for left, and D, dextrous, the Latin for right. Linkstrous and dextrous isomers of these two molecules, all right? And biologically, this is really, really important because almost inevitably, if there's some molecule that can have these two shapes, which bigger, complicated molecules all do, inevitably only one of them will be biologically active and used normally in an organism's metabolism, not the other one. And across all of life, a given molecule is usually used the same way. Oftentimes it's the left-hand version that's used. For some molecules it's the right, but never both. And so it's really important to know what you're dealing with right here for its chemical interaction with other molecules in a living thing. This is terribly important in medicine. Very, very important in medicine and food chemistry. All right, a little bit about those cis and trans molecules, a place that you're familiar with them. You've all heard of trans fats. So here's a trans fat, and here's a cis fat. All right, cis configuration, trans configuration, and you know that one of these is not desirable. I'll leave you a moment to think about it. Take your guess, or state which one is not desirable in your diet. I'll give you a moment. Hopefully you chose the trans, right? You see all kinds of foods that have packaging that say no trans fats, and that's because this double bond right here, so let's go back and look at it over here, this double bond with the long part of the organic molecule running on opposite sides of the double bond is bad for you. This is actually metabolically not good. You need this. You need some of these. These are really, really critical. It's something you can't make. You've got to get in your diet. And you try and take these in your diet and use them for this, but you can't because they're incorrectly put together. And so you end up with a nutritional deficit and also more immediately concerning problems with digestion because you can't metabolize this molecule right here. Gas and stuff like that is a byproduct. Anyway, what we want is fatty acids that have double bonds in them, but we want them to be of the cis variety, like this. And these are the double bonds that we find in oils that come from vegetable matter, what are called unsaturated fatty acids. Unsaturated means there's at least one or two hydrogens that have been, there's at least two hydrogens that have been removed, and when you do that you create a double bond with your carbons, and that little kink we're after right there. All right. Here's another look at enantiomers. So this is alanine, which is an amino acid, and there's dextrous alanine and linkstrous alanine. All right, there are two versions of them. Only the linkstress version, the L version, the left-handed version, and there's chemical rules for why you call this left and this right. It's unimportant for our class. They just exist. But only the L form is used in all living things. So, this amino acid over here is useless. Biologically, you only make these, or things that, living things that make amino acids are only going to make the variety they need. They don't make the variety they don't. But if you synthesize this stuff chemically, you make 50-50 ratios of the two, because that's the way chemistry, in glass works, unless you introduce some sort of mechanism to keep these from being made. And it's also possible, for instance, D-alanine will show up as a nutrient. Many years ago, lots and lots of this D-alanine was used in baby supplements as protein and in pet foods, coming out of a very heavily exporting country. It was used by unscrupulous people. This waste product from chemical industry was added to the material to increase the detectable amount of protein, because it is an amino acid, so it triggers a protein test, and that allowed the material to pass inspection for being brought to the U.S. As dog food or baby food. But in both cases, the material was significantly deficient in protein, and animals and children or babies consuming those products were, at a serious deficit of protein in their diet. Happily, we found out about it and stopped it. But there's an example of where right-handed versus left-handed is important, and in this case, knowledge that this was a chemical waste product, but that could fool detection chemistry, was used to commit a crime, basically. All right. The last important part, and probably the most detail-rich, but the most important, and the thing you've really got to learn well, is you need to memorize these to start with. There's only 7, so it's a small memory job. But you need to memorize the names, shapes, and most importantly, chemical function of these 7 functional groups that you can add to any carbon molecule to alter its chemistry. And generally speaking, the chemistry is altered so that these molecules will dissolve in water. All right, so let me go back a little bit and say what I mean by that. So if we go and look at these real basic chemical, these basic hydrocarbons right here, or these hydrocarbons right here, these are nonpolar covalent bonds. There's no charges anywhere on this molecule, which means this molecule will not dissolve in water. You can put it in water, but it'll group together. So for instance, things made out of non-gaseous hydrocarbons like these, like fats, tend to clump together because they don't dissolve in water. This molecule right here, all of this part right here, is non-soluble. It won't dissolve in water. Other fatty acids will all clump together, and you'll get a droplet of oil in water. And what will happen is this pit right here, which is, these are 2 oxygens right here, and this is a an oxygen hydrogen pair right here. These are going to have charges on them. The oxygens will have a slight negative charge. The associated hydrogen will have a slight positive charge. This bit, which looks like a piece of water really, will dissolve in water. So this end of the molecule will dissolve in water, but the rest of it will not. These guys, all 3 of these, will not dissolve in water at all. Nitrogen is more electronegative, so we'll have a polarity on this bond right here. This will be a slight positive. This will be a slight negative right here. And so this dissolves middlingly in water, and these guys don't dissolve in water at all. When we add the functional groups, all of these, except one, that's this one right here, contain atoms that are more electronegative than their surroundings, and almost inevitably it's oxygen, or it's nitrogen, or in this case right here it's sulfur, and here it's phosphorus. All of these are more electronegative than carbon, and then there's a difference in electronegativity in this case between oxygen and phosphorus as well. So we have very, very polar molecules, and a polar molecule dissolves in water. A polar molecule dissolves in water. This polar molecule here will dissolve in water. This OH and this this carbon-oxygen, that's a polar bond. Slightly positive, slightly negative. This OH right here, slightly positive, slightly negative. That'll all dissolve in water. Same for this one. Same for this one. Only this one doesn't dissolve in water. And this is how it's nonpolar, I can add to the size of an organic a carbon molecule. This is basically a building block to add another carbon, and I can do that over and over and over and over and over again, making an endlessly long chain. I just pop off a hydrogen, attach another carbon, pop off a hydrogen, attach another carbon, on and on. So these all give the carbon compound some sort of chemistry, starting with the fact that all but the methyl group now dissolve in water. And if they dissolve in water, they can participate in aquatic chemistry. And that's required for almost all of the chemistry that occurs in you. The chemistry must occur in water. The chemistry of extracting energy from food, the chemistry of making new DNA, the chemistry of making proteins, all of it requires that the chemistry be occurring in a solution of water. So the hydroxyl group is simply an OH. So R represents the rest of the molecule. Think of R for rest of molecule. Okay. So, here's the rest of the molecule, and then we've got a functional group at the end right here to make this thing, which is called a fatty acid. All right. So R is the rest of the molecule, and here is a hydroxyl group. It's polar. It makes this thing dissolve in water. These are responsible for the classic hydrogen bond that we talk about. And notice this looks like a water molecule, except the H has been replaced by some other organic molecule. Methyl group, as I said, is our one way of increasing the size of organic molecules endlessly. It's nonpolar, won't dissolve in water. A carbonyl group is a double bonded oxygen bound to a carbon. And we can have more organic molecule on either side right here. If the carbonyl is at the end of a molecule, we call it an alkene. If it's in the middle, we call it I forget the term right now. It's unimportant right now, but for those of you from chemistry, you'll recall where the double bonded oxygen is on a chain. It gives us two different classes of names. And then a carboxyl, which is a combination of a hydroxyl and a carbonyl. So at the end of a carbon molecule, the rest of the molecule here, we have a carbon double bonded to an oxygen, very polar, and then a carbon bonded to an oxygen, somewhat polar, and an oxygen bonded to a hydrogen, again somewhat polar. So this is a very soluble bit of a molecule. And when we put this in water, it will lose this hydrogen very quickly. It's the effect of this double bonded oxygen up here, pulls electrons in this direction, and so we break this bond. And the hydrogen, an acidic, missing an electron, so it's got a positive charge on it here, hydrogen is released into water. And as y'all all know, if you release hydrogen ions into water, you're making an acid. That's why this is called a fatty acid. This is a hydroxyl group at the end of a long hydrocarbon, and we've turned it into a fatty acid because it is a fatty hydrocarbon, fatty just being a really, really long linear molecule of hydrocarbon, and there's lots of different choices. And then the acid group at the end makes it a fatty acid. Carboxyl group. All right. If we have an acid, you probably need a base. And that's what the amine group does. An amine group is, here's the rest of the carbon molecule, and an amine is a nitrogen. A nitrogen can make 3 bonds normally. So here's the 2 hydrogens that satisfy its valence chemistry. But when you put it in water, it tends to pick up another hydrogen and give itself a slight positive charge. It shares a bond with another hydrogen. It gets a weak attachment to another hydrogen. Something that takes hydrogens out of solution is a base. So an amine group is inevitably an organic base. This is an organic acid, and this is an organic base. And the place that we're going to see amine groups most notably is in amino acids, which is a carbon molecule that's got an amine group at one end and an organic acid, the carboxyl group, at the other end, and therefore we call it an amino acid. And of course, those are the building blocks for proteins. All right. So there, we've gotten almost all of this taken care of, and a lot of it is stuff that you're somewhat familiar with if you reach through what you've encountered before and kind of formalize this understanding. Then we got 2 different ones. These are 2 functional groups that are very different and do some different things. The first is the phosphate group, And what you want to think here, what I want you to get in the habit of is when you see phosphorus, here's the P for phosphorus, and in this case surrounded by up to 4 oxygens, and that's what a phosphate group is, think energy. This is an extraordinarily unstable molecule that has a high tendency to reorganize itself when in contact with another molecule. And phosphate groups are routinely used to store and transfer energy from one molecule to another. So when you see phosphate groups, the first thing I want you to think of is energy storage and energy transfer. And this is energy storage within a cell. And the molecule we're talking about is ATP, adenosine triphosphate. Tri because it's got 3 phosphates in a row, and adding and subtracting those phosphates is a way of managing energy, metabolic energy within a cell. All right, and it's considered to be acidic. It acts slightly acidic. It tends to release hydrogen ions into solution when you put these in water, and very, very polar, very, soluble in water, extremely soluble in water. Finally, we've got a sulfhydryl group, which is like a hydroxyl, but instead of oxygen and hydrogen, it's a sulfur and a hydrogen. Sulfur also makes only 2 bonds. It's got 2 free spaces in its valence shell, so it makes 2 bonds right here. Sulfur is much more electronegative than carbon or hydrogen, so this will be slightly positive, this will be negative, and this will be slightly positive right here, and that makes this very soluble in water as well. The most important thing that's done with sulfur in with sulfhydryl groups is that you find certain amino acids that have this sulfhydryl group on them. It's only amino acids and therefore only proteins that have sulfur in them in normally. And the sulfhydryl group can bond to another sulfhydryl group. We'll lose the 2 hydrogens, the sulfurs bond directly to each other, and that makes a really, really strong link between 2 regions of a protein, and is one of the bonds that's responsible for a protein's 3-dimensional shape, as we'll see in the next unit. All right. So here's an example of how some of these functional groups, end up making, and replacements make otherwise a relatively simple carbon molecule into a real powerhouse of biology. So here we have a hexane. It's a 6-molecule ring, 6 carbons, except 2 of the carbons have been replaced by nitrogens. All right. And then we've got a carbonyl group over here and a hydroxyl group over here, separate from each other. They're not together, otherwise it would be a carboxyl group. But our carbonyl over here and a hydroxyl over here. Notice a methyl group up here. All right. And these are really polar. This is polar. These are polar. And so because it's polar, it can make a weak bond with a hydrogen over on another molecule. This is called a hydrogen bond right here. Here's another hydrogen bond right here. And this is the bond between one letter of DNA, thiamine, T, and A right here, adenine, which is a double carbon ring molecule with, nitrogens on it. And these are called the nitrogenous bases. Thiamine, adenine, cytosine, and adenine again right here. And this is not adenine, this is guanine. I'm sorry, it's mislabeled. That should be guanine with a G, guanine. All right. And we've got 3 hydrogen bonds very, very classically between cytosine and guanine. So when we look at DNA in the next chapter, we'll see that this is already familiar. 3 hydrogen bonds, which is what ensures that only these 2 guys click together, because they have to make 3 hydrogen bonds. And what ensures that these 2 guys click together is that they can only make 2 hydrogen bonds between themselves. And it's a result of the functional groups. That's the material in this. Let's go ahead and take a look at the study guide. There's a couple of additional things on there. So let's take a look at the study guide. There's a little bit of extra information. There's always a little bit of history that would be useful for you to know. So, as chemists, which really, the ancestor of chemistry, well, there's a lot of them, but, chemistry comes out of efforts by physicians, historically, medical people, herbalists, and in classical sort of medieval literature, alchemists who were always trying to turn lead into gold famously. Well, all of these people started learning various things about what we now call chemistry. Anyway, the foundation of modern chemistry comes, after some, clear prior knowledge was established. For instance, that the four major elements of life, carbon, hydrogen, oxygen, and nitrogen, tend to be represented roughly the same in all living things. So we're all made out of the same stuff in roughly the same ratios, just organized different. The difference between you and I, and then between either of us and a blue whale or a bacterium, is in the specific, molecules and how they're arranged that are made out of these things. All right. Some old history. Berzelius, a late middle-aged, early Enlightenment age chemist, if you will. He wasn't called a chemist at the time. Proposed this idea of vitalism, that organic molecules naturally bred life, and that, life comes from organic material, just it pops out into existence automatically. All right. That there was some magic to, organic molecules, that they had the spark of life in them, if you will. Some German chemists from the late 1700s, early 1800s, Wöhler and Kolbe, worked on the question of organic molecules, and by this time a terrific amount of basic knowledge of organic chemistry existed, but some whys weren't really understood yet. And so at this time it was still considered, when Wöhler and Kolbe were doing their work, that only living things could make organic molecules, and that life was created by organic molecules. That kind of twist back and forth is the basis of vitalism. But they were convinced that you could make organic molecules without life, and to that end, these guys, their work ultimately led to the synthesis of urea, which is an organic molecule, abiotically, without the function of life. Urea is normally the thing that's responsible for uric acid, it's the nitrogenous waste product in urine. It's responsible for the word urine being urine, uric acid. And they proved that you could make uric acid outside of living things, just in a laboratory. Then that kind of popped open a very important question philosophically. Can life arise due to natural processes? All right. And we'll look at that question in various ways as we work through this semester and next semester, Biology 1407. But the answer to that question is, as far as we know, we haven't seen it happen, but chemically it was proved it could happen in theory by a bunch of work, but importantly the work of Urey and Miller, who set up an apparatus that just used inorganic molecules, carbon in the form of CO2, oxygen in the form of water, and nitrogen in the form of ammonia, and a few other things were mixed together in an apparatus that was meant to emulate conditions on very early life, what are called, reducing conditions. And an apparatus just filled with that inorganic material ended up making amino acids, carbohydrates, and lipids from inorganic precursors. And this was just a piece of laboratory equipment, and that proved that it was possible for all of the organic molecules that life is made of to be made abiotically, without life somehow. And there's a terrific amount of support in it out there that this is an ongoing process, and certainly has happened lots and lots in the universe, over time. For instance, we get asteroids and cometary material from space that are rich in organic molecules. There's no evidence of life, just the organic material. All right, we talked about carbon, we talked about four bonds, we talked about chains and rings and branches and big molecules, so that's been taken care of. All right. We talked about hydrocarbons being the simplest of the organic molecules because they're only made out of carbon and hydrogen, and the fact that they have a huge amount of potential energy due to the relative position of the electrons in those non-polar bonds between carbon and hydrogen and between carbon and carbon. We know that the locations of single and double bonds can lead to distinct forms of a given formula. That's what we mean by isomers. And then we had, from that conversation, we talked about structural, which means just same formula, different arrangement, and geometric isomers, where the same formula, different arrangement is controlled by the presence of a double bond or triple bond or multiples of those. And then finally, we talked a little bit about the very important idea of left and right, linksterous and dexterous versions of a molecule, and those are enantiomers. Those are our enantiomers. And for the record, every amino acid has a left and a right-handed version that could be produced, and inevitably we use the left-handed versions of them. And not just us, but all living things. All right. Then we spent some time going over the 7 chemical functional groups, so you must memorize these. These are arranged a little bit different. All of the polar ones are first. So these are all the ones that dissolve in water. The hydroxyl group, if a carbon molecule has hydroxyl groups on it, we're going to call it an alcohol. That's what alcohols are. They've got at least one OH group on that chain. All right. If we have carbonyl groups added to an organic molecule, we're going to typically refer to those things as ketones or aldehydes. That was the term I was blanking on earlier. A ketone being a molecule with the double bonded oxygen in the body of the molecule, and an aldehyde with the double bonded oxygen at the end of the molecule, on one of the end carbons where there's not another carbon on the other side. And if we take hydroxyl groups and carbonyl groups and add them to a molecule, that's how we get sugars. Sugars actually have both of these. They've got one carbonyl and a bunch of hydroxyls, and that's a sugar. Whereas a whole bunch of hydroxyls and no carbonyl, that's an alcohol. And so that's how alcohols and sugars are related to each other. And y'all should know that, right? You gain a lot of calories if you drink a lot of alcohol, right? There's a lot of calories in alcohol. That's because alcohol is a sugar that's been modified by a yeast cell into from a sugar into an alcohol by stripping some high energy bonds off of it. Then we've got that carboxyl, which is a combination of a hydroxyl and a carbonyl combined into one functional group, and that is an organic acid, the acid of amino acid, or the acid of fatty acid. An amine is the inverse. It is basic. It takes hydrogens out of solution and adds them to the nitrogen. An amine has a nitrogen as its central atom in the functional group, and that gives us an organic base. And organic bases, ammonia you think of as being basic. Well, that's a solution that has organic bases in it. That's why ammonia is basic. It's the opposite of an acid. And then we've got things like an amino acid, where we have a molecule with both of these on them at the same time, amino acid, and that is the name for any of the 20 molecules that make up our proteins. Finally, we've got that next we've got that sulfhydryl, which acts like a strong version of a hydroxyl group, and it's capable of forming a cross bond with another sulfhydryl hanging off of another molecule, and that's a strong covalent bond between two molecules using sulfhydryl groups. That's very biologically important. Big deal in protein structure. Then we talked about the phosphate group, and remember the phosphate group, when you see phosphate, think energy. Very electronegative, very, very reactive component, and it's classically used as the coupler for two different chemical reactions. So the way this goes is metabolism. We'll have a reaction that strips energy off of a hydrocarbon or a sugar. That stored energy is transferred to another molecule via the use of phosphates on ATP, and that other molecule can then pay for something that requires energy to be done. That's what we mean by coupled reactions. We have an energy-yielding reaction coupled via an intermediary, a third party, if you will. An energy-extracting reaction coupled to an energy-releasing reaction. an energy-consuming reaction. And it's something with a phosphate that couples those two together. Classically the intermediary. The one you want to think of most prominently is ATP, adenosine triphosphate. Then finally we've got the methyl group, where we can use this to increase the size of a carbon molecule. So that's the raw material for making, methane into a 2-carbon gas. What was it? Pentane? No, pentane's a 5-carbon gas. Propane. I think that's the 2-carbon one. I always forget. Here's an example of those energy transfer molecules. Adenosine triphosphate, which if you pull off one of the phosphates is now adenosine diphosphate. If you pull off yet another phosphate, it's adenosine monophosphate. And all three of these have really big roles in your cells. This is guanosine triphosphate, a related energy transfer molecule, and there's a bunch of others that we'll see in time. But that's the material on the study guide. So the only thing new right here is a little bit about the history of investigation of organic molecules and how we know what we know today. So that concludes your recorded lecture for week 3, Biology 1406.